All right, so what we're going to do today, and probably running a little bit into the next lecture, is give you, in essence, a tour of microbial diversity. Then what we're going to do, and I'll come back to this in a few minutes to give you more details, is spend the next 5 or so lectures discussing different aspects of the diversity of microorganisms primarily. And so if you remember in the first lecture, I walked through a tree of life that organizes the parts of the course. So if we take this tree, this rooted tree that we talked about, we're not going to show on here the mitochondrial symbiosis with eukaryotes. We'll talk about that a little later. If you want to see the diversity of many different groups, it's sometimes easier to draw one of these unrooted radial circular trees. Basically means the same thing. There are 3 major groups here: Archaea, Eukaryotes, and Bacteria. And inside of each of these lineages, there are many different subgroups. And if you look at the organization of the course, on weeks 4 through 5, we're going to talk about plant diversity. That's this branch of eukaryotes over here. In weeks 6 through 9, we're going to talk about this tiny little phylogenetic group here, fungi and animals together, which are related to each other. And then for the next 1.5 weeks or so, we're going to do everything else. That's a lot of diversity to cover. And so if you look at this tree, there's an incredible amount of evolutionary diversity that is not contained within plants, animals, and fungi. There's an incredible amount of functional diversity, of ecological diversity, of organisms that have various impacts on our planet and on human lifestyles and on human health. There's no way that we are going to cover all of this diversity deeply. It's just, there's too much. So what I'm going to do today, and a little bit into the next lecture, is walk you through a little bit about some representatives of the major groups of bacteria, archaea, and eukaryotes. A little bit about their features. And then for the remaining lectures, 8, 9, 10, 11, and 12, we're going to talk about various interesting or important properties of these organisms, about their biology or their evolution or their ecology. And again, this diversity is quite amazing. There are many of the organisms in the rest of the tree are microbial, that is single-celled or small organisms. Microbial basically means too small to see without the aid of a microscope. But there are multicellular organisms in many of these other branches. We will talk about a few of them during the course of discussions. Not all of the rest of the tree is microbial, but the vast majority of it is. So we're going to focus a lot on what we could call microbial diversity. And we're going to talk about a few different sort of properties that many of these organisms have. So some of them are bad. We'll spend part of a lecture talking about the taxa that in particular have negative effects on humans and human health. Go through a few examples across the tree of different versions of those and what they do. We're going to talk about organisms that have mutualistic symbioses with plants or animals or fungi. That is, both of the partners benefit from this interaction. We're going to talk a little bit about what I would call the unusual organisms on the planet. These are extremophiles, organisms that can not only survive, but actually prefer to grow in what we would at least consider extreme conditions. Boiling, acid pools, the bottom of the ocean, frozen environments in the Antarctic, very low or very high pH, and also very salty environments. These are generally called extremophiles because they prefer to grow in these extreme environments. We'll talk also a little bit about how some of these other organisms impact the global biology of the planet, such as global nutrient cycles. And we will also spend a little bit of time talking about many of these organisms and what we sort of don't know about them. And in particular, we'll focus on the cloud of microbes that live in and on people, which we're only just beginning to learn about in more detail. You're covered in lots of microbes. We don't know what a lot of them do, but we'll talk a little bit about how we study them and what people have learned about that collection of organisms. And we'll also talk a little bit about some of these taxa that we leverage in industrial or agricultural processes and make products that we like in some way. So this is the general scheme of the outline for the next few lectures. We're going to do this intro. We'll continue on that intro in Lecture 8 and then talk a little bit more about some other issues in fully sampling the diversity of microbes that are on the planet. We'll spend a lecture basically talking about metabolic and physiological diversity, including some discussion of the Extremophiles. We'll spend a lecture talking about symbioses, which are two organisms that live together. We will talk about cases where one of those organisms negatively affects the other, parasitism, and we will talk about cases where both of the organisms mutually benefit, that is, mutualisms. And then we'll spend a couple of lectures talking about the microbes and the other taxa outside of plants, animals, and fungi that live both on us and immediately in the world around us. So before we talk about the different lineages of bacteria, archaea, and eukaryotes, there's sort of one aspect of their biology that's very useful to introduce before we talk about these individual examples, and that is their ability to carry out diverse biochemical processes. So this is a chart that is basically a chart of compounds. Those are the nodes in this chart, the little circles that I hope you can see there. And then the lines connecting the nodes are examples of enzymes from some organism that are able to carry out the conversion of the products that are represented by the two nodes connected by that line. And if you go out and catalog all the known processes, these biochemical and metabolic processes that organisms are able to do on the planet, converting one carbohydrate or one protein or one inorganic compound into another, and you map, which is not done here, you map the different taxa in which these are found, what you see is a massive amount of the diversity of known metabolic reactions are found in that other part of the tree that I showed you a few minutes ago. The bacteria, archaea, and the other eukaryotes have an immense amount of metabolic diversity. Now this, if you look at the tree, it's not that surprising. They represent in total an enormous amount of evolutionary time, phylogenetic diversity that they have had to evolve different processes. And again, we're not going to be able to cover all of this diversity here. What I'm going to do just before we talk about the diversity of lineages is talk about one aspect of the diversity of processes that are found in different organisms. And this is basically known as their forms of nutrition or trophy. And when people have looked at the ways that organisms acquire their main components for their nutrition, there's been this classification of lots of different aspects of it, but three major components of the nutrition of different organisms are frequently used to classify how organisms basically get their life going from the environment. So we can identify these three major components. One of them is the source of energy that an organism uses basically to carry out their cellular processes. What is the major source of energy that allows an organism to live? And then second is a source of the electrons that they use for reducing power and carrying out processes. So for metabolic processes in organisms, one thing that all organisms have to do is to, in essence, strip electrons from compounds and then use those electrons to carry out various chemical interconversions. And there's a couple of different categories of how organisms get these electron sources. And then the third major category of biological, of nutrition for organisms is where they get their carbon. So for all of us, we need lots of different, metals and minerals and other compounds, but a key aspect of all organisms' lives is where they get their carbon to build up the carbon skeletons that are used for sugars and for proteins and for fats and so on inside their cells. And so if you look at these three different categories, what you can see is if you compare different organisms on the planet, people have classified each of these categories into two main subcategories, different forms that each of them come in. So if you look at energy source, the two major forms of energy that organisms use are divided up into using light. And if an organism uses light as part of its nutrition, they are called a phototroph. So you add this prefix onto troph, and that describes, in this case, the energy source that an organism uses. So there are lots of organisms that have antennae or other processes by which they take the energy of light, they use that energy to excite electrons inside the cell, and then they use those high-energy electrons to carry out various biological processes and make cellular energy out of that. And the alternative is to use chemical energy. So you can take a variety of forms of chemicals, break them apart, and use that breaking of a chemical reaction, of a chemical bond, to generate cellular energy. And if an organism is using chemical energy, they're known as a chemotroph. In light energy, they're known as a phototroph. So we can then go through and look at the electron source and do the same type of thing. There are two different categories here. One of them is organisms that use inorganic compounds as their source of electrons. So for example, some organisms will use water, as we will see a few examples of this both later and in a few other lectures. Water is inorganic. It is used as a source of these electrons for cellular processes and generating cellular energy. And then organic compounds. These are the two main categories of sources of electrons. And if an organism uses electrons, they're known as, from inorganic sources, they're known as a lithotroph. And if they use electrons from organic compounds, they're known as an organotroph. And then the last one of these divisions is for carbon source. And if an organism uses carbon from what are called C1 compounds, that's any compound for which there is a single carbon in the compound. Things like carbon dioxide, carbon monoxide, methane, and so on. Basically, any compound that doesn't already have a pre-existing carbon-carbon backbone, these single carbon-containing molecules. Organisms that use those to build up carbon-carbon skeletons are known as autotrophs. And then the last category are those organisms that get their carbon, in essence, put together, not from C1 compounds, but from compounds that contain two or more carbons. Generally, these are organic compounds that are used, and those are known as heterotrophs. And so we can then classify the biology of different organisms by combining together these different categories in a variety of ways, and I'm going to show you a few examples of this. So the Bacterium E. coli, for example, used as a model organism in laboratories, it's a lives inside of the gut of humans. Generally, it's not harmful, but there are varieties of E. coli that can make us sick. E. coli gets its energy from chemicals. It basically is feeding on sugars and fats and proteins to convert into ATP, cellular energy. It gets its electrons from basically those same chemicals, organic compounds. So it's a chemo-organotroph. And it gets its carbon. It doesn't fix carbon. It doesn't convert C1 compounds into larger compounds. It gets its carbon also from organics. So if you put it all together, it's a chemo-organo-heterotroph. And you will see in the book, and we will talk about in the lectures now, and also when we talk about fungi and plants, and in particular, different examples of these combinations of the different trophies. This is just to take you through an example of it. Frequently, people ignore the middle part, the electron source, and they might refer to this organism, to E. coli, and its metabolism as chemoheterotrophy. So another example, Cyanobacteria, they get their energy from light. They get their electrons from water usually, or other inorganic compounds, and they put together carbon-carbon skeletons from carbon dioxide. So they are known as photolithoautotrophs or just photoautotrophs. Frequently you will hear this type of process referred also to as photosynthesis. So autotrophy is taking these single carbon compounds and building up larger carbon chains. That's also known as carbon fixation, but sometimes it's known as synthesis. So cyanobacterian plants are photolithoautotrophs, but they're also known as photosynthetic. Anybody know what humans are? We photosynthetic? So yesterday I was in discussions with some people who have colleagues who are literally trying to inject, like with tattoo ink, photosynthetic cyanobacteria into humans to see if they can generate some, it's like a gimmick, but generate some photosynthetic energy with cyanobacteria under your skin. It's deranged, but, so humans are chemo-organo-heterotrophs, much like E. coli. So what you will see if we look across the diversity, again, we're not going to go through all the diversity of all these other lineages. There are microbes that have every possible combination of these particular energy, electron source, and carbon source. And also, within each category, there is great diversity. So within the phototrophic organisms, there are many different ways by which organisms can take the energy of light and convert it into cellular energy. There are many different ways that organisms can take inorganic or organic compounds and get high-energy electrons out of them. There is a diversity of ways that organisms can fix carbon, for example. And there's no chance to cover all of this diversity. I just want to sort of hint at it in some of the discussions here. So what I'm going to do for the rest of class today, and then a little bit of the beginning of the next lecture, is now take you through a tour of the diversity of bacteria, archaea, and eukaryotes. They're going to be a small, not a tiny number, but not a huge number of names of the major lineages of these groups. You should learn those names and be able to give examples of key properties of the groups. These are also all discussed in chapter 26 and 27 in the book. We're not going to expect you to memorize or be able to recite species names of individuals unless we explicitly tell you that. But the major groups, you should at least learn their names, which ones go with which domain, so which are bacterial groups, which are archaeal groups, which are eukaryotic groups, and some of the key hallmark features of those groups. So get ready. We're going to go through a lot of different lineages of organisms here. If we look at the bacteria, if you look at them in a microscope, a lot of the time you can divide up the ones you see into three main categories of cell shape. Bacilli are these rod-like ones. Cocci are the circular ones, and Spirilla, there are many different terms used for this, are the spiral-shaped ones. If you see the name of a bacterial species, and also some archaeal species, because some of them have forms like this, you can learn about what the general shape is of that organism, sometimes from the name. So Lactococcus, Streptococcus, Enterococcus, etc., those are organisms that have this type of shape. Bacilli, like Bacillus anthracis, the causative agent of anthrax, has this rod-shaped property. So a lot of information about the organisms is sometimes contained within their Latin name. Now, I'll just go back for a second. This is not a lot of diversity of shapes, and the lack of diversity across the bacteria in their morphology is one of the reasons why sequence information of ribosomal RNA genes and other genes was so essential to classifying and studying microorganisms. We'll talk about this more later. But there are some bacteria that have interesting, informative, and diverse morphologies. This is a giant bacterium called Margarita. It's about the size of the head of a bee. It's actually, I think, originally discovered by Doug Nelson from the microbiology department here. Lives in coastal sediments associated with a variety of other organisms. So generally, bacteria are referred to as microbes. If you take the sort of formal definition of microbe, as in an organism you can't see without the aid of a microscope, occasionally there is a bacterial species that is big enough to see without a microscope, but it's pretty rare. There are bacteria that are multicellular, form fruiting body-like structures where there is division of labor and diversification of function within the structure here. This is an example of an organism called Stigmatella. It's in the group Proteobacteria. I'll show a picture of this later. Another structure that many bacteria form is called a biofilm. It's, in essence, layers of bacteria on top of each other, and in the different parts of the layering, the bacteria acquire different functions. Biofilms are really important in a variety of ecosystems, including inside people. When microbes form biofilms inside, an artery or inside your gut, antibiotics can be very hard to reach to get into the middle of the biofilm, and they can survive for a very long period of time. One aspect of the differentiation between different bacterial groups relates to their cell envelope, the surroundings around the cell. I already mentioned before that all bacteria have this compound peptidoglycan in their cell wall. There actually are two main categories of cell envelopes in bacteria. They're the organisms that are called gram-positive. They're called that because they stain positively with a stain called the gram stain. And they have a membrane around the cell and then a very thick peptidoglycan cell wall outside of that membrane. The gram-negative bacteria have a membrane and then a thin peptidoglycan cell wall and then another second membrane outside of that cell wall. This difference in structure has major impact on how gram-positive versus gram-negative bacteria interact with the world around them. And in particular, of interest to many people is that many antibiotics preferentially work on one of these groups or the other, depending on, for example, how they can either interact with the cell wall or get through the membranes and the cell wall into the cell. So when you have a bacterial infection, one of the first things the people treating you might want to know is it a gram-positive or a gram-negative organism? Bacteria do move. They don't just sit there being sessile. There are many bacterial species that have motility processes. One of the more common ones is to have what's called a flagellum, this long whip-like tail. This, I think, is Salmonella. They actually have many. Some species have just one, others have two. But there's actually bacteria that can tumble, not using a flagellum. There are bacteria that can glide a little bit. There's actually a reasonable number of bacterial species that can move around. The flagellum, if a bacterium has a flagellum, they're all homologous to each other. It's this really cool molecular machine that's basically a rotor and an anchor embedded into the membrane, and then the rotor spins around on this axis, and the flagellum is swinging around on the outside of the cell, and that allows a bacterium, for example, to swim towards certain gradients like light and oxygen and chemicals. They can control the direction they go with these flagella. This is a Vibrio Cholera swimming around. This one's very confused. I don't know if it got like impaled on the slide here, or, but they are remarkably able to swim relative to their size long distances using these flagella. Other bacteria, as we will see when I come to the discussion of the bacterial groups, do use flagella for other purposes as well. So as I mentioned already, the lack of morphological diversity among bacteria in general has led to ribosomal RNA sequencing and other DNA sequencing to be used as the primary means to infer the evolutionary relationships among bacteria. Just like I talked about with Carl Woese doing the initial studies of the tree of life, many people have gone in after that and surveyed a diversity of bacteria, archaea, or eukaryotes even with molecular methods and built finer scale trees. In the book, in version 10 of the book, there are 8 major lineages of bacteria that are discussed. In version 9, there are only 6 major lineages. We're only going to talk about the 6 that were in version 9, because I know some people don't have version 10 of the book. And, but it turns out whether or not they show 6 or 8 major lineages, there are actually hundreds. This is an evolutionary tree of major bacterial lineages. In fact, in this tree, many phyla are collapsed together into superphyla. There are actually hundreds of major lineages of bacteria. There's no way we're going to cover all of them. So we're just going to cover these 6, which are some of the better understood, better characterized, and with known sort of human interest functions. So I'm going to walk you through a little discussion of those 6 lineages. So one of those groups is called Spirochetes. I think most, if not all, of the spirochetes that have been studied have spiral-shaped cells, hence the name of the group. They actually have basically a flagellar-like structure that's embedded inside the cell rather than sticking all the way outside the cell. This is pretty nasty because they can use it to corkscrew their way into tissues and into cells. By spinning around rather than them swimming, they're driving themselves in like a screw. The causative agents of many human and animal diseases are in this group, including the organism that causes syphilis. That's Treponema pallidum. The organism that causes Lyme disease is Borrelia. I think certainly most of the organisms in this group are chemoheterotrophic. There are occasionally free-living ones, but most of them that are known are associated with a host and cause disease in that host. I think I have a little mini video of one of them corkscrewing around. They were, when they were first seen in the microscope, I think they were misclassified, again, because morphology can be deceptive. There are other lineages of bacteria that have spiral shapes that were all lumped together that turn out to be convergent evolution. So the second major group are the Chlamydias. The Chlamydias are all gram-negative, and they are extremely small and live inside cells of other organisms. They are obligate parasites that get their ATP from their host cells. They don't really generate their own ATP. They're parasitizing host cells, and they cause a diversity of diseases. The Classic one is the chlamydial sexually transmitted diseases. They also can cause pneumonia. There's one of them that causes this horrible eye disease called trachoma, leading to a lot of cases of blindness. Because they are so small, and because they live inside the cells of other organisms, prior to ribosomal RNA sequencing, they were generally horribly misclassified. So until the 1960s even, chlamydias, many of them were viewed as viruses. Because they were so small, people couldn't imagine that there could be a bacteria of that size. When you had an infection with something that looked like one of these organisms, you might treat it with the wrong compounds if you didn't know what it was. Another interesting thing about chlamydias is that they can be dimorphic, so they're individual species that actually go through life cycle stages where they change their shapes. And this also led to some confusion where you would find an infection with these multiple shapes in it, and people thought it was caused by multiple organisms, and in fact, it's the same organism in different phases of its life cycle. A third group of the bacteria have a couple of different names that are commonly used. Their formal name for the phylum is the Actinobacteria, but in much of the time they're also referred to as the high GC gram-positives. The reason for this is, first of all, they stain positively with the gram stain. They have that membrane structure that I showed you before, a cell envelope structure with one membrane and then a very thick peptidoglycan cell wall. And the reason they're called high GC is there are actually two different phyla of gram-positive bacteria. This group, the taxa in this group, most of them, if you look at their DNA is heavily biased towards G's and C's in it, rather than A's and T's. So they are called high G plus C, or sometimes you show the ratio high G plus C over A plus T gram-positives. Many of them, when they grow, they have these elaborate branching structures, like here. They look a lot like fungi. Some fungal growth patterns look like this, and again, prior to experimental work, characterization of the inside of these organisms, and in particular molecular work, many of them were misclassified as fungi. Many of the antibiotics that we use in medicine today come from members of this group. So there's a genus called Streptomyces. That's where streptomycin comes from, for example, and many other antibiotics that are like streptomycin come from this group. There are also many pathogens in the group. So Mycobacterium, I think this is a little video of dividing cells of Mycobacterium tuberculosis. Causative agent of leprosy and tuberculosis are included in this group, as well as a variety of other pathogens. The second group of gram-positives are, yeah, question. I think they went out of the frame of the, yeah. I don't think anything interesting is happening to them other than that they just go out of the phase of the camera or something. Oh, wow. I have no idea what's going on here. I think they're probably using some laser that's triggering some fluorescent compound inside of them, and they may be, it's just a side product of the way they're doing it. I think they're accidentally basically wiping them out. The second group of gram-positives are the low GC gram-positives, again, because they have fewer G's and C's per DNA base in their genome. Their formal name is the Firmicutes. Many people call them the Firmicutes, but I refuse to do that. They have a lot of diversity in this group. There are many medically important, agriculturally important organisms like lactic acid bacteria. There are a few individual lineages that actually have lost their cell wall, the mycoplasmas. But I think one of the coolest things in this group, very dangerous but very interesting, is that A couple of lineages within this group have a very interesting specialized reproductive process. And if you watch this little video here, what they do is sort of starts out as a normal cell division type process for a bacterium where they duplicate their genome and then split into two. That would be the normal process. Now watch what happens in some cases, like when the organisms are starving. They go through this process, they copy their genome, they divide, but they divide asymmetrically. Don't know if I killed it. And then the one that is the smaller of these two becomes a spore. It's actually tightly packaged, wrapped up inside of all sorts of protective proteins and carbohydrates. The DNA is packaged very tightly. The cell is dried out, and it is incredibly stable. There are reports of people reviving spores from this group of bacteria that are hundreds of thousands of years old. So that's because they're able to survive incredibly long periods of desiccation and radiation and other assaults. This is what happens in Bacillus anthracis, the causative agent of anthrax, in biological weapons, for example, that have been developed in the letters that were sent to the Senate many years ago. What was sent was spores of anthrax. Those spores are incredibly resistant to just about anything and will germinate when they get exposed to water, like when you inhale them into your lungs. Botulism and tetanus are also caused by organisms that are able to form these spores. All right, two more groups of bacteria here. The cyanobacteria you're probably reasonably familiar with in a variety of ways. If you go out to a pond, in particular a pond that has a lot of nutrients in it, or if you go look at various hot springs or other lakes, you frequently see a film of green on the top of that. In many cases, those are a biofilm, basically, of cyanobacteria. Cyanobacteria actually can frequently form these long filaments, long chains of individual cells. When the cell makes a copy of itself, instead of separating, they stay together, and then you form these long chains. Some of these species are able to undergo developmental processes to form, it's not the same as the spores in the bacilli, but another type of spore, and then they can also form these specialized heterocysts, which are used for nitrogen fixation. Here's an example of a spiral-shaped one. This is Spirulina, which some of you may be eating occasionally when you have some green food drink. Again, the spiral shape looks a lot like the spiral shape in some spirochetes, and it is one of the reasons why the morphology of bacteria is just not that informative for classifying. All members of the cyanobacteria that have been studied in detail are photolithoautotrophic. They contain these complicated internal membrane structures that increase the surface area to allow them to have all sorts of concentrations of light-absorbing reaction centers and also electron transport chains. And as we will talk about later, the plastids that are inside plants and other single-celled organisms, the chloroplasts, are derived from endosymbiotic cyanobacteria. Yeah? A heterocyst is a specialized compartment, basically, that is used for fixation of nitrogen. And the reason they do that is this, the enzyme that bacteria and other species use to fix nitrogen is sensitive to oxygen. It doesn't work really at all in the presence of oxygen. So you need to somehow separate that enzyme away from oxygen that's either in the environment or even inside of the cell. And these heterocysts allow a cyanobacteria to basically create a package, putting this nitrogenase enzyme in the middle of it and keeping the oxygen away from it. And that allows them to pull nitrogen out of the atmosphere and turn it into cellular nitrogen. All right, the last group of bacteria. Oh, yeah, another question. What is the vegetative cells are basically, I think, just resting cells that aren't carrying out a lot of cellular activity, but they haven't changed their phenotype in any dramatic way. They can, but I think they have to be reactivated. I mean, I think they're sort of turned off a lot of their processes. And if I show, just since people may be wondering, if I show a slide and I'm not talking about any of the details on these slides, do a likelihood probability test. I'm going to have a very, very low likelihood of asking you any questions about something that we're not going to spend a few minutes talking about. This is to give you a tour of the diversity, but we'll let you know what parts of this you're going to be needed to learn in more detail. The last group of bacteria that we're talking about here are the Proteobacteria. This is the group that E. coli is in. Incredible diversity of functions. There are photoautotrophic members of this group. There are chemoautotrophic members of this group. There are chemoheterotrophic members of this group. There are pathogens in this group. There are mutualistic symbionts in this group. The ancestor of the mitochondria is in this group. There's just incredible diversity. There are multicellular organisms. Lots of stuff going on in the Proteobacteria. Very, very well-studied group, in part because of the diversity, in part because many of the organisms in this group are associated with plants or animals in interesting ways, so they've been studied in a lot of detail. All of them are gram-negative. And this, I lost. I think this is just some cell division, binary fission of Vibrio Cholera, I think. All right. We're going to switch now to Archaea. On your tour of microbes here. Archaeal shapes. There are archaea that have some of the shapes that I showed you for bacteria. They are less well understood and fewer of them map into those simple cocci and bacilli shapes that I showed you for bacteria. There are some that are really fascinating. This is a square-shaped archaea, archaeal species. Very unusual to form, have your cell, the outer part of your cell form a square. This is, I call this the Mickey Mouse archaea. This is an archaeal species that lives as a symbiont of another archaeal species. Their membranes are actually slightly fused down here. I mentioned before, but just thought I'd mention again, archaea have a synapomorphy, a derived trait that they share in common with these ether linkages in their membranes. Bacteria and eukaryotes have these ester linkages in their membranes. As with bacteria, to build a phylogenetic tree of archaea, the phenotypes have not been that useful. Most of the phylogenetic trees that we have are based upon ribosomal RNA or other molecular data. The book, in the tree used by the book, highlights 2 lineages of archaea. In the text, they actually briefly discuss 2 other lineages that are very, very poorly understood. We're just going to focus here on these 2 main lineages of archaea. But again, if you look at this tree here, there are actually at least 10 major divisions of archaea, probably 20 plus phyla of archaea. We're just going to talk about 2 of those groups here. The Kren Archaeota, most of them are, you could call them extremely extremophilic. They are both thermophilic, that is, they like heat, and acidophilic, that is, they like acid. So many of them live in places like boiling acid pools that you can see in Yellowstone National Park, for example, or other places where there's active geothermal processes, or in drainages of mines where there's acid coming out associated with the mining process. An organism called Sulfolobus is very well studied in this group. It's basically become a model organism in this group, and you'll see occasionally a lot about it. This one, ferroplasma, can even live at a pH of 0. It actually prefers to live at pH 0. That's basically more acidic than hydrochloric acid. The second group of archaea are the Euryarchaeota, and there are 2 main kinds of Euryarchaeota that I will tell you about. One are called methanogens. The name describes what they do. They generate methane as a byproduct of their metabolism. They take CO2. They are autotrophic. They use CO2 and they convert it into methane. The methanogens across the planet have a major role in global climate change. Methane is an important greenhouse gas, and these methanogens generate a lot of it. They live in the guts of ruminants, for example, and so the belching and farting of cows releases a lot of methane thanks to methanogens. We actually have methanogens. Many people, not all humans, but many have methanogens in our gut. It's not clear exactly what they're doing there, but many of us have them. And you can diagnose who has them with various features, but we'll talk about that some other time. And then the last group of archaea we will talk about is another lineage of the Eukaryotes, and these are the halophiles, organisms that thrive in high salt conditions. They grow in places like evaporating lakes or evaporating ponds, or humans create such places called solar salterns to evaporate water and recover the salt that is left over. And if you go by the Great Salt Lake or by the South Bay, where there are a lot of these solar salterns, you see as they get higher and higher salt concentrations, the colors change. That's different organisms colonizing them at the different salt concentrations. When you get to the really high salt concentrations, the halophilic archaea take over. One thing that organisms in this group have that's really interesting is in natural environments and also in those human-constructed environments, as the water evaporates, eventually you desiccate. The water availability becomes very, very low until the environment completely changes. And these halophilic archaea are incredibly resistant to desiccation. Not quite like the spores of those bacilli, but there are many reports of people recovering halophilic archaea from very, very old salt crystals. And if you're interested, you can go to the beauty store or to the Nugget or to any other place that sells natural salt or pink salt, and you can grow the halophilic archaea out of those samples. The halophiles generally, there are no archaea that are known to be human pathogens. So you'd probably, wait a sec. You don't have to worry about what is in those pink salts. But just one last thing about these halophilic archaea. Many of them have a light-absorbing pigment called bacteria rhodopsin that they use to generate cellular energy. They generate ATP via this light-absorbing compound. They don't fix carbon. So they are an unusual example of photoheterotrophy rather than photoautotrophy. And we will continue in the next lecture on microbial eukaryotes. Did you get all that? And be overwhelmed. There are other toxic influence.
It's take it easy, a little easier now for the rest of the day, right?
I always have to put notes for where I stopped for each section.
Peter's here.
Yeah, I saw. He likes to learn.